Now, in the fifth step, the pericranial division of the pro-embryo takes place to form 16 cell structure, and this division is taking place in such a manner that we will get, or the embryo will receive one outer cell and one inner cell. So this is the first, second, third, fourth and fifth. So fifth step is pericranial cell division of pro-embryo, and this pericranial cell division of pro-embryo takes place in such a manner that one outer cell and one inner cell is formed. Now, what about these outer cells? These outer cells act as a dermatogen. So, outer cells act as a dermatogen. Gen means formation. Dermato is for skin. So, dermatogen will form the skin, that is the outermost layer, and we know the outermost layer of skin in case of plants is called as epidermis. So, dermatogens will give rise to epidermis. Right? Now step number 6. So this is step number 5. One outer, one inner cell. Now cells of hypophysis divide to form an 8 cell structure. Hypophysis cells divide to form 8 cells. Right? These 8 cells are arranged as a ball or a bunch of cells, but for our convenience we will show them separate cells, because each and every cell is responsible for the formation of some of the other parts of the plant body. So hypophysis cells, that means cells of hypophysis, will divide to form 8 cells. Earlier it was only one hypophysis, right? The one which is located towards the chalaza end or in the contact of pro-embryo. So one will give rise to 2, 2 to 4, like that. So these are the 8 cells of hypophysis. Now let us discuss about the fate of these hypophysis cells, again with respect to or in contact with the microphylla or chalaza end. So 4 hypophyseal cells which are located towards the microphylla end, or you can also say located towards suspensor, that means we are talking about these 4 cells, will give rise to root cap and root tip. So the A part, that is the 4 cells towards M end, that is microphylla end, will give rise to root cap and root tips. Right? Now what about the upper 4 cells which are located towards the chalaza end, or they are in the contact of pro-embryo, whatever you want to say. These cells will give rise to root cortex. So 4 cells towards C end, that is chalaza end, will give rise to root cortex. Now, if like we have divided 8 cells of hypophysis into 2 halves, 4 towards microphylla or towards suspensor, 4 in the contact with the pro-embryo or chalaza end. Similarly, we can divide this pro-embryo also. That means pro-embryo half towards chalaza end, half towards microphylla end. So pro-embryo also, that means half cells of the pro-embryo which are located towards, half of the pro-embryo cells which are located towards the C end or M end, they will also form different types of structures. Right? So half cells which are located towards the microphylla end, that is M end, they are going to form hypocotyle. So they will form hypocotyle, right? And half located towards chalaza end will form plumule as well as cotyledon. So half cells of pro-embryo only located towards C end, that is chalazal end, will form this cotyledon and plumule, right? So we have divided the cells of pro-embryo also. So this is the root cap and root tip, this is the root cortex. Now we can write hypocotyle, cotyledon as well as plumule. That means this part, that is half cells towards the micropylar end, that means this region will develop hypocotyle and the upper part towards chalazal end will develop plumule as well as cotyledons. Now what happens is, again then the maturation stage will come, the heart shaped embryo will also form like that. So let us draw one diagram which is the most common diagram, right? This is the embryo sac, and here let us say the two cells, that is the initial one, basal cell and the terminal cell, right? And the upper one, the part is located with the cells of, all these are the cells of endospermic cells, right? All these are endosperms. So this embryo sac, within the embryo is formed within the embryo sac, this suspensor pushes the embryo towards the endospermic region, so that embryo should provide or embryo should receive the nourishment from the endosperm. This is what we call it is the globular form of embryo. This one is globular form of embryo. Now what happens is, when the hypocotyle, cotyledons and plumule form, the structure of embryo changes. Now how the structure of embryo changes? This is the first structure, that is the globular form of embryo. Now again this is the embryo sac. We can say that hypocotyle will form in this direction. So let us say from this region the hypocotyle forms. So this is the mass of cells which is hypocotyle. Now, and before hypocotyle, this cotyledons and plumule also develop. So let us say this is the centralmost part which will give rise to plumule, and the cotyledons are still remaining. So this is the region of, we can say this is the region of cotyledons, and the entire cell mass is accumulated in between, right? So this is the plumule, and the two cotyledons are arranged in such a manner that it will provide us some heart-shaped structure embryo. So these are, you can say, the two cotyledons. So this is heart-shaped embryo, where this is hypocotyle, these are the cotyledon, and this is the plumule. Now, if again the cell division will continue, then what will happen? Instead, in the embryo sac, the space will be very less. So what happens is cotyledons bend, because hypocotyle will develop, cotyledon, this plumule will also develop, and one more thing, that radical will also develop, epicotyle will also develop. So these structures will develop from this middle region. So when they are growing or dividing, they are maturing in this region, what happens is these cotyledons will receive some sort of pressure. When they receive pressure, they are pushed and they are bent. So when they bend, they will form structure something like this. Right? The cotyledons are arranged in this manner, and the root as well as shoot system also develops. Right, and this is called as horseshoe shaped heart. So earlier it was globular form of embryo, Then cotyledons develop as a heart-shaped embryo. Then embryo when pushed, they will develop the horseshoe shape. Right? Now one more thing regarding the structure of the embryonic axis we are going to discuss. Within the structure of embryonic axis, so say this is the structure of embryonic axis. This upper part will be called as the plumule. You can see from here also, which will give rise to shoot system. Lower part is composed of radical, and the radical will give rise to root system. So plumule for shoot system, radical for root system, and the two cotyledons. So these are two cotyledons. Now hypocotyle as well as epicotyle. The structure which is present between plumule and cotyledon is called as, this is the region of epicotyle. So epicotyle is, epi means above, cotyledon, cotyli for cotyledon. So between plumule and cotyledon. BW means between plumule and cotyledons. Now between radical and cotyledon, the structure which is located is called as hypocotyle. Hypo means below. So hypocotyle is located between or below cotyledon, radical and cotyledons. Right? So initially it was the globular form of the embryo. Then it changes to heart-shaped embryo and then to horseshoe-shaped embryo. So this was all about the development of embryo in case of dicots, that is dicotyledonous. In the next part, we will discuss about the various steps involved in the development or formation of monocot embryo.